specific molecule or category of molecules found in a membrane.
Mr. Horner, please call the main office. Mr. Horner, please call the main office.
What makes up the bilayer? Phospholipids is one of your answers. That is a macromolecule, a phospholipid.
Oh, the first essay question.
So if you choose phospholipid, we need to say something about the structure and something about the function, correct? Structure and function. So what can you tell about the structure of a phospholipid? By the way, you did a DLE where you told me the structure of a phospholipid.
Fatty acid.
There is a hydrophobic section. What's the hydrophobic section? What are those tails made out of? So we have two fatty acids in our phospholipid that make up the tail.
It's funny because Chris is fat.
Narrated by Chris.
I know this was a question on a quiz, wasn't it? What's the part at the top?
Phosphate head.
What's that made out of?
I should do that. I could freaking do that. I could set it. No, I can't.
What connects them all? What's the molecule in the middle that connects the tails to the head on a phospholipid? Nope. Glycerol. There it is. Who said that one? Nice call. That is glycerol. So one of the simplest things you can say about the structure of a phospholipid is it's made out of two fatty acid tails, a phosphate head connected by a glycerol molecule. You get all three of those, you clearly have explained the structure of a phospholipid. Now, what else could we say about the structure of a phospholipid? What can we say about the nature of this phosphate head?
Hydrophobic.
It is hydrophilic because what is its more important chemical nature? It is polar. There we go. Polar is a structural thing. All right. In the actual answer key, I was looking for polar for the head, which means what am I looking for the tails? Non-polar. So now we've got two structural things we can say. All right. Phosphate head that is polar. Two fatty acid tails that are non-polar, connected by a glycerol. That's two different structural things. Now let's get to function. What do these things build in membranes? They form a lipid bilayer. All right. Now, what is the purpose of that bilayer? What is its overall function?
Separate the inside from the outside.
To separate things, to act as a barrier. To separate the inside from the outside. You are correct. So, it is this non-polar fatty acid tail that is a barrier. And what is it a barrier to? What does it prevent from crossing the membrane? Barrier stops hydrophilic molecules from passing. Okay? Hey, what could you say about the head that is part of its function? What does it interact with well? It is hydrophilic. What does it interact with? Water. So what portion of the membrane is the head? Is it inside or outside? It is the top and bottom surface. Yay! So phosphate head polar. Hydrophilic equals interacts with water. Does that sound like enough for a phospholipid? Yeah. So again, what are the structural things we talked about here? We got it. What part is polar?
Phosphate.
The phosphate head is polar. What part is nonpolar? Fatty acid tails. What are the three things that make up this phospholipid? Phosphate, glycerol, fatty acid tails. Two of them. Two fatty acids. So that's structural stuff. Functional stuff. What's the overall purpose of a bilayer? Act as a barrier to stop what kind of molecules from passing the membrane? Hydrophilic molecules can't pass. And then, of course, the function of the hydrophilic end of this, the reason why there is a phosphate here, is it allows it to interact with water and form the surface of that barrier. All right? That is one of your four options for a type of molecule. Can somebody name another macromolecule found in?
Cholesterol.
We have cholesterol. So what can we say about cholesterol as a structure?
It decreases fluency in the membrane.
The most basic thing about cholesterol, which category of molecules is it in?
Steroid.
It is a steroid. That's a subcategory. Go even bigger than that. Yes, it's a steroid. But what else? Is it a carbohydrate? No. What category? Dun, dun, dun. It's a lipid. Ding, 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 ding.
What? What's so funny?
Now you could describe it as having a four-ring, a four-carbon ring structure. But just identifying as a steroid and a lipid should work for the structure part. Now what's the function of cholesterol in a membrane?
Like the fluidity of the.
Does it make it more or less fluid?
Less.
Less fluid. Function reduces fluidity of membrane.
I didn't say that word. Fluidity.
We know who's getting it. No, you're
And it does that because it has a funny shape. That funny shape of the cholesterol bangs into all those phospholipids and prevents them from moving very easily. Think of it as kind of like, a rock in the road they have to get around. All right? Besides reducing fluidity, what does it do for membranes at cold temperature? Prevents it from
freezing.
solidifying, or freezing. All right, that's two. We had phospholipid, we had cholesterol.
You said prevents what?
Prevents it from solidifying or becoming solid. So we had phospholipid, cholesterol. Two possible molecules left. Yeah.
Oh, transport protein or any type of protein.
Proteins, right? It's a fluid matrix. That matrix is a variety of proteins. What are the functions of those proteins in the membrane? There are only 6 listed in your textbook, so tough to figure one out. We talked about proteins today in the membrane. In today's lecture, there was a protein in the membrane. What was it? It was a receptor. There you go. All right, so proteins can act as receptors for detecting things like chemical signals. What else could proteins in the membrane do?
Transport.
Transport what? Yeah, transport molecules across the membrane by a process called... It could be active transport or facilitated diffusion. That's correct. So we could have active transport pumps. We could have facilitated diffusion channels. By the way, can you name any?
Sodium potassium pump.
The sodium potassium pump is a protein found in membranes. Ding, ding, ding. So you could identify that as an example. But really, just stating the general, they act as transport proteins. They can be pumps or channels. That works as well. Okay? Hey, what's the structure of a protein? Since we have to define structure? It is what of amino acids? So meaning what? It is a polymer of amino acids. That works. It's a polypeptide. A chain of amino acids. That all works. A polymer made of amino acid monomers. Any way you want to get across the concept that a protein is a lot of amino acids hooked together, that should be your structure point rather easily. Last molecule in the membrane or on the membrane? Carbohydrates, and one of those carbohydrates attached to by the ER and the Golgi. Starts with a P. What are carbohydrates attached to on the membrane? They're attached to proteins.
That's a first.
So in fact,
Stop.
What is saccharin?
I sit right there.
¿Qué tiene la clase? Isn't it? That's another.
Right there. He's not even done.
I need to go get my grade keys.
Oh yeah.
So, I guess the post is at 5. So we have
So-
until then to make the posters.
these carbohydrates that are attached either to proteins or they can even be attached directly to phospholipids, they exist as polymers, meaning they're chains of, what do we call the individual carbohydrates? Monosaccharides, yes. These are chains of monosaccharides, so you could call it a polysaccharide. And what is the function of these carbohydrates? Anyone? Anyone? Bueller? Bueller?
Receptor.
Got your ID card?
Receptor.
They are for cell identification. They are a part of you identifying your body as your cells. So you will have, so carbohydrates, they're part of cell-to-cell communication, and they're part of identifying self. When your immune system needs to know if something is a part of you or is foreign, the immune system will have cells that come along, and they will have receptor proteins that bind to these carbohydrates. If the receptor fits to the carbohydrate, then this cell is safe. If the receptor doesn't fit on the carbohydrate, then the immune system will attack and kill this cell. That's how you keep you, and the foreign bad guys out. Anybody who does not have the right carbohydrates, and therefore the right ID card, dies. Yeah. Yep. Either directly on the phospholipids or on proteins. Yes. And by the way, on which surface here? Are these going to be on the inside facing the cytoplasm or the outside facing?
Outside.
Yes, these are outside only. Hey, remember what you call a protein that has carbohydrates attached to it? Starts with a gly, right? Yeah, that is a glycoprotein now that it has carbohydrates attached to it. All right. Am I going to be on YouTube later? Man, it's like college.
Can they say
All right, so to reiterate, once again, function of the phospholipids. Function of the phospholipids as a barrier to prevent what kind of molecules from passing? Hydrophilic molecules, right? Will not go through. Function of the cholesterol. To lower membrane fluidity by just getting in the way. Function of the proteins, whole bunch of things. Receptors, cell recognition, transport proteins, structure, lots of functions for the proteins in the membrane. Functions of the carbohydrate, cell-to-cell recognition or cell identification, yes. Any questions about that? So pick your favorite 3 out of those 4. Describe those 3 and only those 3. Do not bother describing the 4th one, it won't be graded.
Only 3.
I'm like a writer.
That was
Is that what it did? Let me see your notes, Alina. It's like a typewriter.
What I said is a second. I might draw in something else. And then label that polymer amino acids. Okay? Hey, so hey, now with the first essay, there's really only 2 parts, aren't there? So guess what each part's going to be worth? Half of the essay. Typically, the essays have about 12 points as a maximum, even though your high score is a 10. So if 12 is the total number of points, how many do you think are being part A? 6 points. Wait, 6 points possible. We're describing 3 molecules and we say something about structure and function.
Yes.
2 features times 3 molecules. 2 times 3, 6. So if you say 1 successful thing about structure for each molecule and 1 successful thing about function for each molecule, should you get all 6 points? Yes. Okay. You should. Now part B. Explain how the sodium-potassium pump works. Again, didn't we do this
Yes.
in a DLE?
Yeah.
Didn't you have this on a quiz? Go back and look at that. That was this question in different words. So you're going to explain how the sodium-potassium pump works again. And again, 6 possible points. Gee, there were like 6 steps in the process, weren't there? Huh. Wow. Does that make sense? Yeah, and on the quiz, you had a maximum score of 6, didn't you? Yeah. Yes.
I thought it'd be like this too.
The molecules. There were 4 possible molecules. Protein, carbohydrate, cholesterol, phospholipid. Here are your 4 choices. 6 points. So pick your favorite 3. Pick your favorite 3 out of those 4 and describe something about their structure and something about their function. Just that easy. All right, do we need to go over how the sodium potassium pump works?
No.
Thank you for that. You're disappointed.
Sorry for anybody who did. Go do it on your own. Good luck with that.
Yeah. Cholesterol, first of all, it's a lipid. It is a steroid. It has a characteristic steroid backbone that is made out of 4 rings of carbon. That counts as structure. All right. You could also, I suppose you could call it nonpolar. There's another major element of its structure, nonpolar. All right. Let's move on.
I need your email.
Send him to the
You notice in the second essay, once again, we're back on structure and function, correct?
Yeah.
So now we've got a 3-part essay. If it's full of 12 points, probably how many points in each part? Probably 4. The first part asks you to talk about 2 organelles, and for those 2 organelles, say something about structure and function. Wow, 2 times 2 equals 4 once again. So you're going to pick your 2 favorite membrane-bound organelles, successfully say something about their structure and something about their function. Let's pick some organelles that are surrounded by membranes. Golgi, there's one.
Crap, that's gonna be maybe October.
What can you tell me? What can you tell me about the structure of Golgi? Flattened sacs that are surrounded by
Whoa, Alina knows. Cisternae.
word starts with an M, ends with an membrane. Yeah, these are membrane-surrounded flat sacs. Or membrane-bound.
Membrane brown sacs.
Is it cross-connected?
Organelle,
cross-connections. Function.
Modified sort. Modified sort.
Modify, how's it, what is it modifying?
Protein.
Modify proteins by adding
Carbohydrates.
carbohydrates. Add carbs to proteins. That's the modify. Then what's it going to do with them? Sort and ship to. Sure. Don't overcomplicate things. Everybody can remember carbs, right? Yes, you could also say they add lipids. They can make lipoproteins as well. That's the glycoproteins. But again, let's keep it simple. Anyway, modify by adding
Sort and
carbs,
ship.
sort the proteins, ship the proteins to their destination. That was pretty simple, wasn't it? Okay, another organelle. ER, we got 2 kinds, right? We got rough and smooth. Okay, we'll go rough. Tell me something structural about rough ER.
Cisternae.
Take that exact same explanation, membrane-bound, flattened sacs that are cross-connected. And there you got ER. Anything special to rough ER that make it rough?
They have proteins on the outside. Proteins.
You understand my shorthand there? So the rough ER would be membrane-bound, flattened sacs that have cross connections and there are ribosomes on the outside. Sounds like a good structure? Yeah, that should cover it. Function. What do these guys do to proteins? I should hear an F word here. Fold and modify by adding carbohydrates.
You're wrong. I was wrong. Chris was wrong.
Because he's wearing the Mexico shirt.
And there's another major important part of the cell that the rough ER generates. Starts with a mem. Yeah, the rough ER makes membranes as well.
Oh my god.
New winded. So there you go. You got 2 things you can say about function. Is it always good to say 2 things when you really only need to say 1 here? Yeah. If you can add like just half a sentence and cover a second function, that makes sure that if you have a complete brain screw up and you get the first one wrong, you've got that backup function there to help out. Yeah. So like if you had said that the rough ER detoxifies proteins, which it doesn't do, does it? I mean, detoxifies toxins, which it doesn't do. But then you say it is also the place where new membranes are made, you'd get the point for the function.
Oh, nice.
Okay? If you had said that the rough ER is, membrane-bound sacs that are not cross-connected, but then you went on to say they have ribosomes studying the outside, the ribosome thing gets you the point. Okay? Yeah. How many what? Same as last time, 22 and a half minutes. Hey, how many of you were a little bit rushed last time?
What?
Yeah. Hey, but then again, how many of those people who just raised their hand got like a 9 or a 10? Yeah, you were writing a lot of stuff. It was good stuff. There were lots of good answers last time. Not going to complain there. All right, so shall we take some other options on organelles?
Six or seven.
Let's, hey,
No, it's just then that's bad quality.
could we at least just identify the other membrane-bound organelles so you can at least recognize them? So, looking for something that starts with a V. Structure for a vacuole? 3 words. Membrane-bound sac. There you go.
It's just a sack.
Same question. Boy, isn't that easier? And what does this membrane-bound sac do? What do they store?
Good job, Kelly.
What? Food, waste? There we go.
Mr. Kazire, please report to the main office.
Another one.
Breakdown.
I'm on my way.
Breakdown of the.
Oh no, another organelle. Come on, storage of food waste, that's plenty. What? Smoothie R. We can say smoothie R. So the structural description is going to be everything you said about rough ER, but forget the ribosomes, right? Membrane-bound flattened sacs with cross connections for smooth ER. Function for smooth ER?
Lipid synthesis.
You can synthesize lipids, yeah.
You're only picking two.
What do you store in the smooth ER in muscles? Calcium storage? Hey, what's getting stored in there? And what's being detoxified in there? Well, what organ detoxifies, I should say? Yeah, liver uses detox. Let's move on. Beyond smooth ER. What else?
Wait, are we only picking two of them? Are we picking two of them?
You only need to pick 2.
Okay,
That's
so
for
can
the
we talk
first
about the rough and then the smooth ER?
I'm not sure if rough and smooth are 2 different organelles on the answer key. They might be, but they might be one.
That's for the first
So
one,
in
right?
fact, I would pick one and I would pick something else. Yeah. You could have mitochondria. Yep. Anything you know about the structure of mitochondria? Bean shaped. How many membranes? 2 membranes.
Whoa.
Anything special about the inner membrane? The inner membrane is folded. Hey, anything else you know structural about a mitochondrion? What's inside it?
It's DNA.
What's inside it that might have information?
DNA.
It's got DNA. What helps take that information to make proteins? It has ribosomes. Okay? Say all that. Hey, what's a mitochondrion do?
HATP.
I should hear an R word. Respiration, which means we're making what molecule? Another organelle. Lysosome. There we go. Structure of a lysosome? Yes, it is a membrane-bound sac. We'll take this and we'll just copy it right there. Hey, if it were me answering this question, I would be describing vacuole and lysosome. Membrane-bound sac, membrane-bound sac. Although membrane-bound sac, hey, what makes a lysosome slightly different? Membrane-bound sac with what inside? With a low pH or an acid. So acid-filled. Membrane-bound sac. Oh, we're going to call that an MBS. Function?
Breakdown of cells.
Yeah, got me a more advanced word than breakdown. Like, you know, with a D. Digest, there you go. Hey, anybody know the name of a reaction where you're digesting using water? Starts with an H. Hydrolysis, there we go. That's all the stuff.
No sé si te va súper bien, no te mucho ahorita, Huh? Like you use the super beast on this and much trust and do really shitty on the SS. Can't fives. You know?
Intracellular digestion. Yeah, if it were me, I would go vacuole and lysosome because they're both membrane-bound sacs. Lysosome adds acid. The functions are really simple. Vacuole, store food and waste. Lysosome, break down old cell parts, organelles, proteins. Intracellular digestion. Yeah, that's quick and easy. Membrane-bound, flattened sacs and cross connections. Man, too many words.
That's true.
That was only for part A, right?
Yeah. Hey, there's at least 2 more membrane-bound organelles you can discuss. One is a plant-only thing. Chloroplasts. And function for that? Starts with a photosynthesis. Hey, how many membranes are on the chloroplast? 2, just like a mitochondria. What else does it have inside like a mitochondria? DNA. Ribosome. All right. You could do chloroplasts and mitochondria. They're both easy. They're both double membrane surrounded bean-shaped organelles. They both have circular DNA inside and they both have ribosomes inside. The structure is easy. One of them does respiration, the other one does photosynthesis. That's piece of cake stuff. One more organelle that's similar in shape and size to lysosome. Peroxisomes, there you go. Which is, once again, peroxisome structure would be a membrane-bound sac filled with peroxides, not acids, peroxides. And do you remember something that peroxisomes do? Hey, the one thing I want you to remember that peroxisomes do is they break down fatty acids so that they can be used to produce ATP. All right. The peroxisomes don't make the ATP. They're usually right next to what organelle it's going to make the ATP? They're next to mitochondria, so that they can break down the fatty acids, send those products to the mitochondrion. The mitochondrion uses it to make ATP. All right. So lots of choices for Part A. No shortage of membrane-bound organelles. Part B. This is what draw, label, and explain we did. For comparing and contrasting prokaryotes and eukaryotes. DNA, cell wall, ribosomes. Is it okay? And pick 2. And it says, so hey, once again, it's asking you to pick 2 things and describe its function and how they're different in the 2 types of cells. 2 times 2 is 4, right? So for each one, get 1 function and then explain 1 difference, and you've got all 4 points. So what's the function of DNA? It carries genetic information. Yeah, that wasn't hard. How are they different in the 2 organisms? Guys, play handsy later. Ooh, location. All right, so where is the DNA in a eukaryotic cell? In the nucleus. Where is it in a prokaryote? In the cytoplasm is all you need to say. Yeah, you could call it a nucleoid, but who wants to be that complicated? All right, so they are different in their location. Eukaryotes have DNA in a nucleus, prokaryotes do not. It's in the cytoplasm. Anything else different about their actual structure? I'm looking for a shape here. Which one has circular DNA? Prokaryotes have circular DNA. Our DNA is not in circles. What is our DNA?
Double helix. The double helix.
Don't ignore it. That's all double helix.
Oh.
But their double helix wrap around so that the beginning meets the end. Yeah, ours is linear. That's correct. Our DNA is in strings. Their DNA is in loops. And, yeah, that's good enough. Right? Okay, cell wall. Purpose of a cell wall? Protection. What else?
Maintain cell shape.
Shape. Cell shape is a cell wall function. I'm looking for another one.
It keeps it together.
Prevents it from exploding in a hypotonic solution or due to water gain. I'm looking for another basic one. It's the reason why my desk doesn't fall over. Support, yeah, okay. This is nothing but cell walls, you know that? Wood is just cell walls from dead plant cells. That's all it is. All right, which means, hey, what molecule, if this is plant, what molecule is it made out of? Cellulose, that's correct. Eukaryotic cell walls are made out of either cellulose, or what are the fungus ones made out of? Chitin. All right, that's the eukaryote side. What's the prokaryote side? What are prokaryote cell walls made out of? Peptidoglycan. Everybody say peptidoglycan. Pepto-Bismol. All right, last, ribosomes. Function of ribosomes? What about proteins? I'm looking for two words for the function of a ribosome. Make proteins, there you go. So if you're looking for a short answer, ribosomes make proteins. How are they different between prokaryotes and eukaryotes?
Their location.
What's different about their size?
They're smaller and
Prokaryote ribosomes are smaller. Eukaryote ribosomes are bigger. Remember, make both halves of the comparison. Don't just say prokaryote ribosomes are smaller. Finish it out by saying eukaryote ribosomes are bigger. Anything else you know about prokaryotic ribosomes that allows us to kill them?
Oh, they're allergic to, more sensitive to antibiotics.
Prokaryotic ribosomes are sensitive to antibiotics or can be stopped with antibiotics. Eukaryotic ribosomes are not sensitive to antibiotics. Again, make both halves of the comparison. Do not make just one half of that comparison. Both statements. You got to have both statements or you don't get the credit. All right. Anything else different about ribosomes? Yeah. In which organisms?
Mitochondria and chloroplast.
In eukaryotes, ribosomes can be found attached to some membranes, the rough ER and the nuclear envelope. Whereas in prokaryotes, as in eukaryotes, for both, in prokaryotes, the ribosomes are only in the cytoplasm. So eukaryotic ribosomes can be in cytoplasm or attached to two different membranes. Prokaryotes do not have those membranes. They can only be found in the cytoplasm. All right. Sound easy enough? Yeah, part B that's pretty simple. I expect you to do well. All right, hey, part C, endosymbiotic theory. What is the endosymbiotic theory?
Mitochondria came before.
Blah, 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 blah,
Plus
blah.
one.
So, what did mitochondria and chloroplasts, we believe, used to be? Free-living prokaryotes. You want to get that phrase in there? Free-living prokaryotes. You want to get across the concept that they used to be independently growing organisms.
Which ones? Eukaryotes and what?
We're free what?
The chloroplasts and mitochondria used to be free living or independent prokaryotes. But they then moved into larger eukaryotic cells and became internal symbionts.
What was it? Can you repeat that?
So that later they moved into eukaryotic cells and became internal symbionts or endosymbionts. Symbiont? It's actually in the question. Symbiont?
Do we have to know how they got into the cell? Do we have to know how they got into the cell?
Internal and then
What? How I said it. But in the essay, you just say, do the phagocytosis. So then it kind of like,
All right. Endosymbiont, something that is alive, that is together inside. Yeah. So again, bless you. The endosymbiosis theory says that chloroplasts and mitochondria used to be free-living, or you could say independent, prokaryotes. They then moved into larger eukaryotic cells and became internal symbionts, or you could say endosymbionts, or you could just say simply started living inside of eukaryotic cells, to use simpler English. Yeah? Larger cells, right? The going theory is that the nucleus had already formed by then, so we could call them eukaryotes.
can we say invaded or?
Yeah. You could say plastids and mitochondria, yeah, because a chloroplast is a type of plastid. Yep. Now, the rest of the points in this question, you get 1 point for saying what the theory is, which is what we just went over. The rest of the points here are for explaining the evidence. All right, and it does say A, but I think you can get points for more than one. So what evidence do we have that mitochondria and chloroplasts are similar to prokaryotes? Yeah.
Fungus and animal cells.
Hmm?
Fungus and animal cells.
What about them?
They,
Mitochondria.
I forgot.
Are you talking about the reason why mitochondria we think came first?
Yeah.
Yeah, we're looking more for the evidence that mitochondria are similar to prokaryotes. Brian? They both have circular DNA, and they both have a single DNA molecule. Those are both prokaryote characteristics that, of course, you could have talked about in Part B. That is one piece of evidence. Yeah? They divide by simple division. They both divide by a splitting process. They do not divide by a mitosis process. Yeah?
Similar ribosomes.
They have similar ribosomes in that they are small and sensitive to antibiotics. That would be 3. They have ribosomes that are small like prokaryotes and are sensitive to antibiotics like prokaryotes.
Similar protein.
And they have similar proteins in what part? Their inner membrane have similar proteins to each other. Yeah, those are the 4 pieces of evidence from the book.
What's the last one?
Their inner membrane proteins are similar. Yeah. You do not have to draw, not necessarily, no. But again, drawing can help make your explanation, make sure your explanation is correct, if you label. Why? So that you'll do these, yeah. Again, hey, it is a saying in sports, you should be more afraid of your coach than the other team. Right? You should practice harder than you have to play, so that when you get to the game, you feel you can Right.
What am I videotaping? It's been 45 minutes.
All right, hey. Shh. All right, one question.
Does mitochondria the only one that has circular DNA, or is it both?
Mitochondria and chloroplasts. Everything we said about mitochondria was true about chloroplasts as well.
Singular or circular?
Circular.
Both. They have one DNA. It is circular. All right, hey. For the third essay, the very first thing it says, graph the data on the axes provided. So what do you need to put on this graph? Tell me what's going to be around the sides of the graph. What are you going to write on the x-axis?
Molarity of sucrose.
You're going to put molarity of sucrose, right? And what are the units for molarity of sucrose? Molar. You're going to put both of those on the x-axis. What are you going to put on the y-axis? Percent change in mass. What are the units? Percent. What are you going to put over the top of the graph? A title. What would be a fine title? Molarity of sucrose versus percent change of mass is a wonderful title. Then you're going to plot these data points. How many different symbols do you need to use to plot these data points? No, there's only one set of data here. Yeah. You just, dots will do. All right, now once you draw those dots, what are you then going to do on the graph? You're going to draw what kind of line? Yeah, draw a best fit line. Okay. And let's see. Da, 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 da. And it says, find the apparent molar concentration. How will you do that? Where what crosses what?
X-axis.
Where the best fit line crosses y equals 0, that value of x is your apparent molar concentration. All right, so graph dot 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 0 on the y-axis, line here. You're just going to go. Equals, and then, you know, whatever it is. That's all you got to do. Whatever value of x this is, that's the molar concentration. Please don't leave off the unit. Don't give me a pure number. So what's the unit going to be? Yeah, m for moles.
Capital.
Okay? So in that part A, you're going to get a point for setting up the graph properly, meaning scaling your axes, labeling the axes, putting a title on. Hey gentlemen, we're having a review session. So again, you're going to get a point for scaling your axes correctly. You're going to get a point for labeling the axes, putting a title on, and plot. You're going to get a point for actually plotting things correctly, and you're going to get a point for figuring out what the correct concentration is. Yeah. It's not asking for slope. Nope. All right, part B. What are the components of water potential? Pressure and? Oh, come on, Glenn, you know these answers. See ya.
Yeah.
So. Jeffrey. So, what's that one?
Water potential.
Water potential overall. What's that one?
Water.
Pressure potential, and that one? Solute potential. So your very first sentence, water potential is made up of pressure potential and solute potential. Then, why is water potential important for moving of water in plants? Second sentence, what direction does water move?
High to low.
Water moves from high to low water potential is your second sentence. Then you're going to explain how water moves through plants.
So water potential or pressure and gas? In this section of Gabby's video, Dr. Leete erases it on the whiteboard and then writes
Well, Ricky
In this
wants to
section
tell
of
me
Gabby's
something.
video, that is a mushroom.
Is that a camera?
Camera.
It's a camera phone.
No,
It's a phone
it's
with a
strictly
camera.
just a camera.
Why don't you have like a...
This is like a flip, except it's Kodak.
Pressure.
So it's not a flip, it's a Kodak. You silly.
Oh, and that's when we talk about
Said-
radiation.
Water potential.
Pressure, really.
Because, look at it.
Hey, so, ladies and gentlemen, what do we call it when a plant loses water through its leaves? That's transpiration, yeah.
Transpiration. Xylem. There's just so
Wait,
information in this unit.
that is a tree. That's a tree.
I thought it was a mushroom. Yeah, me too.
It's a tree.
Well, because the bulb at the top.
You don't like my ball of leaves at the top?
No, it looks like
It's just one big leaf.
Yeah, but there
Gotta be.
probably be green mushrooms, maybe.
Leaves.
There, does that help?
Yes.
Thank you, okay.
It's a tree.
Hey, so, shh, shh, shh. Let's think this through. Water from the xylem is escaping into the air. Does that mean the xylem is going to be just as full as before, more full as before, or less full? Less full. If it's less full, what does that mean about the pressure?
Goes down.
It will go down. If the pressure drops, what happens to the water potential? It goes what? Water potential and pressure potential go together. They go up together, they go down together.
Decreases.
Pressure drop equals lower water potential. Which means down here, what do you have? If this is lower, higher water potential. In what direction is the water going to flow? There you go. See, it was just that easy.
Oh, it does. It only flows in one direction.
To put this in sentence form, as a plant releases water by transpiration, the pressure in its leaf xylem will go down. This lowers the water potential in the leaf xylem.
Play back. I will have that sentence down.
Again, as a plant releases water from its leaves by transpiration, water release by transpiration. It's okay, you'll see it on, what, YouTube later? We'll
Ask
just
Gabby.
upload to, you know.
Gabby Science, guys.
By the way, you're going to call this Tom Talks, okay? These are not TED Talks. These are Tom Talks. All right. Again, as water is lost from leaves by transpiration, the pressure in the leaf xylem drops. This lowers the water potential in the leaf. Because water potential in the leaf is now lower than water potential in the root, water moves up from the root to the leaf.
Because water potential.
Again, because water potential is lower in the leaf than in the root, water moves from the root up to the leaf.
Higher to lower.
Boom.
Again, that's all the plant is doing, is it's setting up a water potential gradient where there is high potential in the roots and low potential in the leaves. That causes water to go upward. Yeah. Solute potential and pressure potential.
Maybe a couple hours, I don't know.
Yeah.
Tom